Unit 2 time again earlier. So Unit 2 Paper 2 Biology, June 2017. Here we go. Question 1. A simplified summary of the stages in glycolysis is given in figure 1. So what we see in here? We see now we start off with glucose. This is what what you get in your sugars that you eat. And it turns into fructose biphosphate, then into triose phosphate, which is a 3-carbon sugar, and ultimately into pyruvate. Now remember, pyruvate is what goes into the Krebs cycle so that it could make citrate, and that kind of initiates that whole cycle here. So hopefully we don't have to talk about that here. So what they're asking me to do is label A, B, C, D, E, and F. And you can see the labels already there, but let me explain what's going on so that you'll know, what to do in case the question come out a little different. Alright, so first of all, you'll notice, well, the whole process of respiration, glycolysis is the first step. The whole process of respiration, right? And let me show you a little flowchart here. So this flowchart here will kind of go into overview of the whole thing. So cellular respiration start with glucose. This is what you get from your food. Glucose is broken down during the process of glycolysis. Now this process produces a net gain of 2 ATP, the energy currency. This occurs in the cytoplasm, by the way, not the mitochondria. Now all of this here would initiate the Krebs cycle through something called the link reaction. Now remember that the pyruvate is what helps establish this, right? The Krebs cycle is used in aerobic respiration, starting with pyruvate as well as there, and remember that comes from glycolysis when the glucose is broken down into them 3-carbon sugar compounds, and ultimately one will end up initiating the Krebs cycle. Now the Krebs cycle occurs in the mitochondria, not the cytoplasm. And this has a net yield of 2 reduced NAD or NADH, 2 ATPs, 2 reduced FAD or FADH2, and this then initiates the electron transport chain to make 36 overall ATP. Is also used in anaerobic processes, but we're not going to get into that there. So that's a basic overview of cellular respiration of the step-by-step process. So let me look at an overview of glycolysis. So if you look here, we have a 6-carbon sugar. That's glucose, 1, 2, 3, 4, 5, 6. And this eventually converts into a compound called fructose biphosphate. And you see that this is basically glucose with 2 phosphate groups added to it. Where do those 2 phosphate groups come from? Well, ATP has 3 phosphate groups, ADP has 2. So one of these is lost from each, so a phosphate group is lost from each ATP here, and that's why I end up with 2 here. Why do we have to do this? This is because the fructose biphosphate is unstable, and what has to happen is that this 6-carbon sugar has to break down into 3-carbon sugars. So this will, think of it as a break in half, and you end up with 1 here and 1 here. There's only really one we have to look at, and that is this 3-carbon sugar here with this phosphate group attached to it, and this is called glyceraldehyde 3-phosphate. But we could call it something simpler, and we could just call it triose phosphate. Why triose? Because triose has 3 sugars. That means 3 sugars, because 1, 2, 3 with a phosphate group attached to it. Easy. Now what happens here now is that this triose phosphate eventually has to, think of it as it has to lose this phosphate group now and turn into a sugar called pyruvate, which is just 1, 2, 3 sugars. How does it do this? Well, it requires an enzyme that is called a dehydrogenase. So a dehydrogenase enzyme, if you think of it, it is a hydrogen removing enzyme that will have to come into play here. NAD is something called a coenzyme, and think of that working together with dehydrogenase. What does it do? NAD is like a battery that has to be charged. Right now, NAD is uncharged. How does it get charged? It gets charged by accepting a hydrogen. So NADH is when it becomes charged. It's kind of like you're catching a ball. You're catching a little ball of hydrogen and you're holding it in a glove. So NAD accepts a hydrogen and basically is dehydrogenating something else. And this phosphate group now gets attached from this ADP to this ATP, and then again here. This whole thing happens twice, so you actually end up with 4 ATP down here. But since you had 2 ATP being used to start this process to turn to to allow the phosphorylation, which is just the addition of phosphate groups of glucose to fructose biphosphate to make it unstable enough now, that used 2 ATP here. So if you think about it now, if it's using 2 ATP to produce 4 ATP, well, how much net ATP we getting? Well, we making, in the end, we getting a net total of 2 ATP. And that's that process there. So once again, you just have to fill in the blanks here. A lot more happens than what you're seeing here, but this is what will work for now. So the next question here is explain the significance of the process labeled X in figure 1. So if you look here, where X is where glucose is converted to fructose biphosphate. The difference is that this fructose biphosphate has 2 phosphate groups attached to it that make it a little unstable, and that allow it to split into these 3 carbon sugars down here, triose phosphate. So if you look at the answer, ATP is required for the phosphorylation, just a big word that means addition of phosphates, to glucose in order for it to become fructose biphosphate. And biphosphate because it has 2. ATP converts ADP during this process. The fructose biphosphate eventually was unstable, so it must be split into 2 pyruvate molecules, which are needed for the next steps of cellular respiration. Well, actually here, I should say it must be split into 2 triose sugar molecules, which converts to pyruvate, which is needed for the next steps of the cellular respiration. Okay, so the question here now, explain the significance of the process labeled X in figure 1. And if we look up here, this process X is the conversion of glucose into fructose biphosphate, which is basically the addition of 2 phosphate groups to it. And if you look back at this diagram here, you'll see that glucose gets converted into this, and the only real difference is that it has 2 phosphate groups attached to it, and those come from the ATPs here. Why does it do this? So it could be unstable to turn into triose phosphates, which is 3 carbon sugars, which eventually turn into pyruvate. And that would be the essence of our answer here. So ATP is required for the phosphorylation, just a big word that means addition of phosphates to glucose in order for it to become fructose biphosphate. It becomes unstable there, and the fructose biphosphate eventually split into 2 triose sugar molecules, the glyceraldehyde 3-phosphate, right, G3P, which converts to pyruvate needed for the next steps of cellular respiration. B. Mitochondria play an important role in cellular respiration. In the box below, draw a label diagram to show the detailed structure of a mitochondrion. You don't have to title it. And then they say on the diagram, use label arrows to identify 4 key substances of cellular respiration that move out of the mitochondrion. Okay, so what we're going to put on the screen here is a diagram that you could make a drawing of in the mitochondrion, and you'll see some labels and annotations, but you really just have to label it. And I'll put substances that are going into the mitochondrion as well as out. In the case of this question here, you just have to put the 4 that you're seeing that are coming out as arrows. Alright, so let's look at C. Ecosystems are dynamic in nature. Figure 2 illustrates the changes in population sizes of an agricultural pest and its predator over time, following the introduction of the predator species. So the predator is acting as a biological control here to help reduce the pest population. Now remember, the problem is that sometimes when you introduce a predator, is that predator itself could overwhelm the other species in that food web, in that ecosystem, and the predator could become a pest too. So you want the predator population to also go down. But let me look at what's happening here. So they say describe the changes in the two populations during the period, and this is for a total of 3 marks. So you want to have at least 3 points here. So if you look at the beginning of the graph, the pest population is high, the predator population is low, and that's basically what you're seeing in the response here at the beginning. Predator population low, pest population is high. Because now the predators would be feeding on the pests, the predators would have enough energy and food available to start to reproduce, and their population will start to rise. So as their predator population start to rise, the pest population will start to fall because they're getting predated upon, right? So predator population rises, pest population falls. But as the pest population starts to fall, there's less food available for the predators. So as the pest population falls, so does the predator population. You'll notice that the pest population is always would stabilize in the end to be higher than the predator population, because there must be more food than consumers. So in the end now, both predator and pest population stabilize, with the pest population being slightly higher. Not too bad. We will look at the next one. Question 2. We have a nephron question here. Figure 3 is a stained section through the medulla of the kidney, showing cross sections through some functional regions of the nephron and associated ducts. You'll see in here that this is a section through the medulla, or the inner part of the kidney, and you'll see 3 parts labeled: 1, 2, and 3. So just to tell you what the labels are here, 1 is actually the thick region of the loop of Henle, 2 is a collecting duct, and 3 is the thinner region of the loop of Henle. So they say identify the cross sections for each. So as I just said, 1 is the thick region of the loop of Henle. How do you know that from the diagram? Now you'll notice, and maybe I could zoom up a little bit here just to show you. You'll see that the cell borders are actually quite difficult to distinguish. Now look at the collecting duct, for example. You could actually see clear divisions in the cells. So you can see it here, for example, but in these ones here, in the loop of Henle, they are a little more difficult to distinguish. All you can really see it here, they are more cuboidal looking cells, and that's how you're able to tell that this is the thick section of the loop of Henle. The collecting duct is the label for 2, and how are you able to tell? Well, you'll see inside here, this is the lumen of the collecting duct, and the cells actually bulge towards the lumen because they would have to facilitate reabsorption of water through the aquaporins, which are water-filled protein channels in the collecting duct. The cell borders are also easier to distinguish. So once you identify each region and you give at least one reason why, you should be fine to get your 4 marks. Alright, so part 2, comment on the main function of the region labeled 3. So 3 is the thin section of the loop of Henle. What this would do, I'll put up a little diagram to just explain this a little further, but it's just 1 mark. What this does is that it creates a high solute concentration of sodium and chloride ions. Now remember, a high solute concentration means a lower water potential. And remember, for osmosis to occur, water flows from where you have a higher to a lower water potential. So this is basically used to reabsorb water, and that should be enough for you to get 1 mark. Alright, so part 3, they say in the box, make a detailed drawing of the cross section labeled 2. I'll put up the drawing here. You don't need to label or anything, but what you want to have is the cell division, the nuclei being shown, the cells and the cell borders being shown, and just, overall neat drawing. There's no need to provide title or magnification. Simple 3 marks. Okay, so let's look at B. Figure 4. shows the results of some experiments in which the effect of the closure of stomata on transpiration rate in a plant is studied. 2 series of experiments are conducted. So you have one in still air, as what you're seeing here, and one that's happening under windy conditions. So let me just look at the graph just to make sense of it. You will see on the y-axis there's the rate of transpiration, and on the x-axis there's the stomatal aperture, basically how much is the stomata opening, like the diameter of the opening of it, and that's in micrometers. Now if you look in wind, and in wind there's a lot of air moving, of course, and it's moving around water vapor all over the place. In wind, you can see that the relative rate of transpiration remains pretty high. It's not linear, but it has a much steeper gradient than in still air. So in still air, usually like when no wind is blowing, we could see that the relative rate of transpiration is much lower, but the rate also plateaus. Very early on, it actually plateaus somewhere around or close to the halfway point, about 5 to 10 micrometers. It actually begins to stop increasing steadily. The question here is asking now, with reference to the structure of stomata, explain the nature of the relationship between stomata and transpiration. It's 3 marks. First, you explain what is stomata, and those stomata are pores mainly found along the leaf's lower epidermis. They are surrounded by guard cells. Those guard cells will then open and close, basically depending on if they are turgid or flaccid, and that controls the aperture or the size of the opening of the stomata. When the guard cells are open, transpiration rates increase, thus allowing the diffusion of water vapor out of plant leaves. So when those are open, water vapor flows out and transpirational pull and so on occurs. The guard cells regulate the rate of transpiration in response to various environmental factors. A good example being temperature. In high temperatures, you typically will have a lot of water flow through the plant to cool it down. Part 2, using the data given in figure 4, that's the graphs, compare the general trend in transpiration rates in still air and under windy conditions. So we just talk about that. In still air, the rate of transpiration increases gradually with stomatal aperture, while in wind there's a much steeper increase in the rate. The overall rate of transpiration is lower in still air when compared with wind, and in wind the rate of transpiration increases steadily with opening of stomatal aperture. So again is what you're seeing here, right, where it begins to kind of level off. So it doesn't matter how big the stomata opens. What happens here is that the relative rate more or less remains constant, or there's only very small increases as it goes along. Whereas when wind is blowing and moving, water vapor around, there's a much higher rate as the stomatal aperture increases. Suggest an explanation for the results obtained for the experiment done under windy conditions. Well, in windy conditions, regions of humidity, or those that are saturated with water vapor, they are carried away from the leaf, and that increases rate of water diffusion out to the leaf. That should be fine to get 1 mark. Question 3. The percentages of the total deaths in 2012 that were attributable to alcohol consumption for different age groups in 2 regions are provided. And here we could see different age groups, basically 15 to 19 going all the way to over 80. And we have the percentage of total deaths due to alcohol consumption. And we have a comparison of the Americas and Europe. The first part here is asking us, on the grid provided, plot a bar chart of table 1. I'll put it on the screen here. You could either draw this, as the Americas and Europe side by side, or you could draw them stacked one on top of the other. It's probably easier to draw it side by side. Now looking at the data here, they said state 2 conclusions that can be made based on the data provided in table 1. And if we just look at it here, we could see that generally the people that are below 50 years old, they have a much, in both Americas and Europe, they have a much higher death rate due to alcohol consumption or any alcohol attributed deaths. In Europe, it is higher overall, and as the after 50, basically as they as the people get older, there's less and less alcohol attributable deaths. You can see like it's in the 20s in Europe and it drops below 20 here, is over 10 in the Americas, but after 50 years old it drops, below 10. So those are the points you want to make there. Alcohol attributable deaths of all age groups are higher in Europe compared to the Americas, and the number of alcohol attributable deaths are highest in younger age groups and decreases in older age groups. Discuss 2 social consequences of excessive alcohol consumption. I have 3 here, but you could have much more. Alcohol addiction can lead to neglect of personal responsibilities of work, leading to possible joblessness and ensuing poverty. Well, basically, you could lose your job if you drink too much, and then you won't make any money. And then all kind of other things has happened. Alcohol abuse affects concentration, inhibition, so it reduces the inhibition, and sensory perception. This could lead you to be kind of overconfident, and as a result, now this could result in in violence, increase in public altercations or even domestic violence, increase in drunk driving and car accidents. And so these 2 would work, but the third one, alcohol abuse leads to neglect of personal responsibilities at home. This includes financial decisions. It can lead to isolation from the family, stigma, possible domestic violence. But it could also exacerbate mental health issues due to that isolation, such as depression. Any one of these could work, but again, you want to stick to social consequences, not biological consequences. So here you're going to be talking about ADH and thing here. Describe 5 risk factors that promote the onset of type 2 diabetes. Type 2 diabetes is the one that comes later in life. It is due to insulin resistance mainly. It is not due to non-production of insulin. Remember, that's type 1. This is the one where you do produce insulin, but your cells become resistant to it. So, first risk factor here is obesity. Visceral fat around the organs leads to insulin resistance because you have inflammation and damage to those tissues. Sedentary lifestyles, basically lifestyles where you're not doing much, is very inactive. It leads to buildup of fat in the body, which of course contributes to obesity. 3, a diet of refined sugars and high in fats, especially coupled with a sedentary lifestyle, contributes to obesity and type 2 diabetes as well. Type 2 diabetes becomes more common as age increases and is more prevalent in families that have a history with the disease. It is also more prevalent in certain ethnicities. So any 5 of these would work. The concentration here is basically around general health and family history. Question 4. Light intensity and carbon dioxide concentration are 2 factors that can limit the rate of photosynthesis. Discuss the concept of a limiting factor in photosynthesis. Okay, so what a limiting factor is basically, think of it like this. If that factor is not present, the process will not occur. So if you don't have light, photosynthesis will not occur. Even if you have water and carbon dioxide and the correct temperature and thing, light in this case is the factor that will actually limit the rate of photosynthesis. It will not, if it's not there, it will not occur. So a limiting factor decreases the rate of photosynthesis if the factor is not present or if the factor is in low concentrations. So if you have a low light intensity, that will result in low rates of photosynthesis. If the rate of photosynthesis increases as the intensity of the factor increases, the factor is still considered limiting because the more, if you're increasing it more and more, and the rate of photosynthesis is going up more and more, well, really, what you're doing is that it's still limiting because the more you increase it, the higher the rate is going. So it's still a little confusing, but think of it like if you're using light as an example again. If you have a bulb and you're making that bulb brighter and brighter, basically increasing the light intensity, and you feel the plant photosynthesizing more, but the light is still a limiting factor there. Now eventually what will happen is that the rate of photosynthesis will become constant even if that light bulb or that the light intensity is extremely high. Well, at this point, the factor is no longer really limiting. It might be some other factor being limited. It could be the amount of carbon dioxide. It could be the number of chloroplasts. It could be the amount of water being available. But at this point here, it basically plateaus. And I'll put up a graph to just show you where light would be a limiting factor and where it will not be a limiting factor. Alright, so you have a kind of big question here. Using your knowledge of the light-dependent and light-independent stages of photosynthesis, explain how light intensity and carbon dioxide act as limiting factors. So with light intensity, at low light intensity, the rate of photosynthesis is low, as only a limited number of electrons can become excited. And remember that there will start off that whole process called photophosphorylation. I ain't gonna go into that here, but think of the electrons as acting like little balls on a pinball machine that, you know, initiating reactions throughout the chlorophyll. So light stimulates those electrons, and those electrons, think of it as they allow photosynthesis to happen. So if you have more electrons stimulated, more photosynthesis happens, greater the rate. As light intensity increases, the rate of photosynthesis increases linearly as more electrons become excited. More light, more electrons excited. More energy is also being made available for the production of 3-carbon sugars during the light independent stage. This is during the Calvin cycle. At high light intensities, all the electrons are stimulated, so the rate of photosynthesis does not increase further. So at a point now, you get all the available electrons, all of them stimulated. If you keep adding more, you can't really stimulate anymore because all of them already stimulated, right? So it's kind of like you keep adding more by not really making a difference. In fact, you might actually damage the chlorophyll as a result. This is basically, think of it like it reaches a saturation point. So remember, at this point here, the light will no longer be a limiting factor, because if you keep adding more, it will not limit the process at all. Alright, so carbon dioxide concentration. Carbon dioxide is needed for photosynthesis, and eventually that carbon dioxide would get reduced to form sugars. So at low CO2 concentrations, the rate of photosynthesis is lowered. And of course, it's the same thing there. If you increase the CO2 concentration, you would increase the rate of photosynthesis. So why does that happen? Well, that increases the activity of the enzyme called rubisco. Rubisco is kind of like what kicks off the whole photosynthesis process during the light independent stage. What that does is that it helps fix that carbon dioxide for the production of 3-carbon sugars. Now at higher CO2 concentrations, CO2 is not really a limiting factor anymore. Again, it's the same, you keep adding more, it's not going to make a difference. And that means that the rate of photosynthesis no longer increases. So this here was 6 marks. And you want to have about 3 points here. You want to state what happens at low intensities. Why is that a limiting factor? Like, what effect does it have? And at what point, does it stop becoming a limiting factor? Alright, so B, microorganisms are primarily responsible for the cycling of nitrogen in the biosphere. With reference to the main processes in the nitrogen cycle, outline 4 key roles of microorganisms in the nitrogen cycle. So there's a pretty basic one here, and this is I think it might be 4 marks, 5 marks. What you want to do is name the 4 types of bacteria and just give a basic overview of what each one does, just linking it back to the nitrogen cycle. So the first one we have here is during nitrogen fixation. Nitrogen fixing bacteria are found in the nodules of roots of plants, for example legumes. And what those do is that they convert nitrogen gas from the atmosphere into a more usable form, which are nitrates and ammonia. Ammonification basically occurs when dead organic material is decomposed by bacteria or fungi, and then converted into ammonia as a result. That dead organic matter is turned into ammonia. Nitrifying bacteria convert ammonia to nitrite and nitrate. So these are in the soil. And so this is called nitrification. Ammonia and nitrite, right, has to be converted in, again, a more usable compound called nitrates. Nitrates are used for growth and so on. So nitrifying bacteria does that. And finally, any nitrates that might be in the soil has to be converted back into nitrogen gas. So denitrifying bacteria does that. This is called denitrification. This basically takes the nitrates in the soil and converts it back into nitrogen gas so that it can go back into the atmosphere and then get reused and get converted back into nitrates and so on from by nitrogen fixing bacteria. I'll put up a little diagram here of the entire process so you can have a little visual to understand. Okay, so 5, we have a beast of a question here. Anything with action potential could, cause a lot of stress. So explain the mechanism by which a nerve cell membrane is able to conduct an action potential and give an analysis of 2 factors that affect the rate of conduction. So what I'm going to do is I'm going to record something else. I have a little simulation that is being used to explain all of this here. So you could take note of everything here, and I wouldn't repeat it, but you could take note of the 5 points that I made there. And you'll see the simulation now. Let me try to explain this action potential thing here. This is a neuron, or this is the axon of a neuron. This is the membrane of the axon here. Let me take a look at what's going on. Closer. You're gonna see a bunch of red dots. These red dots, these are positive sodium ions. You're going to see some green dots. Those green dots are positive potassium ions. You're going to see now that there's a negative charge inside here and there's a positive outside here. Why? Because there's more sodium, there's more positive charges outside here and there's less negative inside here. So this is because of sodium-potassium pumps. Sodium-potassium pumps, for every 3 sodiums that come out, only 2 potassiums come in. I say they are not actually depicted here. I'll put up a little picture just to show you. And this is why inside here would have less positives. This is why inside here is more negative. So this is what you call the resting potential, right? So this is the point at which the axon is not stimulated at all. So what you'll see is that the resting potential before any impulse comes is about minus 70 millivolts. So inside here is slightly negative compared to the outside. Now let me look at what happens when an action potential comes. We're going to look at this graph too, right? So it looked like things happen a little faster than we wanted. But let me look again, and then I'll explain what's happening. And look at the charges here too. Well, first we have to understand was what here. So remember I said the red dots is sodiums, the green dots is potassiums. Both of those are positively charged. We have these channels that remain open. These are called leak channels, right? So these basically, the green ones allow potassium to move via the membrane. The sort of orange ones allow sodium to move. These closed ones, these are gated channels. These channels open in response to changes in voltage and so on. And right now they are closed, so nothing could flow through them. But when they get stimulated by an electrical impulse, look at what happens here. So we're going to play this in slow motion and a way to observe what happens. What's going to happen is the inside here is going to become positive. Why is that? Because when this electrical impulse comes, this is actually going to open all of these sodium channels, the red ones. And when these sodium channels open, there's a bunch of sodium ions gonna start rushing in. So when these sodium ions start rushing in, inside here is gonna become negative and outside is gonna become positive. That's exactly what you're gonna see happening there. Now after that happens and in here becomes positive, when you feel breathe for some moments, it's going to become positive. And what's going to happen now is that these potassium, these green ones, these potassium channels are going to open and they're going to let potassium flow out. And that there is going to cause the outside, right, to become positive again. So you're going to see positive turn into negative, back into positive on top here. Now let me look at it again. So you're looking at slow motion. So you're going to see all the sodium rushing in, the gates open. Now all the potassium going to be rushing out. Gates close. And it actually takes a little while to go back up to minus 70. Now we look at the graph now. That point there, where down here becomes positive inside the axon because all the sodium flows in, that is called depolarization. Right? That's called depolarization. And when depolarization occurs, that's when you're seeing this rise in the graph here until it reaches into the positive region. Now, coming here, this one that's coming down, this is called repolarization. This is where all the potassiums are coming back out. So you're going to do this again. And this is going to be depolarization, is where you see when it's flowing in, and all these flowing out now, this is called repolarization. So flowing in, depolarization, flowing out, repolarization. But something happens here, is that it ends up going a little more, it overshoots. So it ends up being a little more negative than it's supposed to be. So a little more flow out than really supposed to come out. And as a result, now that's called hyperpolarization. We don't need to talk about that too much here. But what happens during that point, hyperpolarization, right? So once again, depolarization and repolarization. And you see that it becomes a little more negative than it's supposed to be. This allows a period of time before a next depolarization process could occur. Because they see in here that it's climbing little bit as it coming back up to go back up to minus 70 millivolts. This period here, this is called the refractory period. It's basically like a little lag time before you could have a next depolarization occurring. And hopefully you understand. You can watch our next YouTube video. So the next part of the question here is to give an analysis of two factors that affect the rate of conduction. Well, those two factors are what you see in here. Basically the diameter of the axon and the concentration of sodium ions outside of the axon. Well, axons with greater diameters allow faster conduction of impulses. What you would have seen there is that on the membrane itself there's a lot of pumps and a lot of channels, and having a greater diameter, and think of it as having more surface area too on the membrane, would allow there to be more pumps and more leak channels, so that this entry and exit of ions can occur at a faster rate. Now you need sodium ions to sort of initiate this action potential. So having a higher concentration outside would increase the rate of conduction, because they would be able to pass through the pumps and through the channels at a greater rate. Okay, so B, outline the general principles of hormonal action in animals and discuss the role of hormones in the regulation of blood glucose concentration. So here I just have a description of what hormones are. So hormones are chemical messengers secreted by endocrine glands. Endocrine ones are ductless, which means that they secrete right into the bloodstream. They are transported to target cells to allow performance of specific actions and usually have a role in homeostasis. Examples would be insulin and ADH. So let's look at regulation of blood glucose concentration. Well, there are two main hormones involved in this, that is insulin and glucagon involved in this. And when homeostasis has to occur, we have to understand that what we'll have is what we'll call a set point or a norm. And this set point, right, this could either go higher or lower than normal. And what will have to happen is that some kind of negative feedback mechanism would have to bring it back down to the set point, or bring it back up to the set point. So the one that involves if the blood glucose level, which is typically around we could say between 80 and 100 milligram per deciliter, if that goes too high, especially like if when you eat sugars, you eat very sugary food, it's gonna climb because it's gonna get absorbed into the bloodstream. This is the one where you're gonna need insulin to bring it back down. And when it goes too low, we say that you didn't eat any sugars in a while or deprived of it, this is where you're gonna need glucagon to bring it back up to the set point. That's the basic behind it. And now we're gonna get into what happens. Well, when the blood glucose level increases above the norm, above the set point, beta cells in the pancreas secrete insulin into the blood. What insulin does is that it activates enzymes in the liver to facilitate uptake of glucose from the blood. It also activates enzymes necessary for the synthesis of glycogen. Glycogen is the storage form of glucose from the glucose. So glycogen, remember the liver stores a lot of glycogen. And for when the blood glucose level falls below the set point or the norm, alpha cells from the pancreas release glucagon into the blood. Glucagon would bind to the receptors on liver cells. Insulin doesn't bind to the receptors, right? Glucagon binds to receptors on liver cells and stimulates the breakdown of glycogen to glucose. That glucose would then be released into the blood. You could also have the breakdown or the conversion of other non-sugar substances into glucose. So for example, fats can be converted to glucose. That is a process called gluconeogenesis, which just means the formation of new glucose. Those of you all who are familiar with the ketogenic diet, which is basically consume a lot of fats and a little bit of sugar, a lot of those fats get broken down into glucose eventually. So this could also happen with the help of glucagon in the liver itself. Popular topic here, question 6. Vaccination is regarded as a form of active artificial immunity. The way to distinguish between the terms active immunity and artificial immunity. So active immunity is, think of it like this, wherever though kill it is make you stronger. It is basically when you get exposed to an antigen or a pathogen and you form antibodies against that antigen, and you're able to fight off a future infection faster. So active immunity occurs when a person's immune system has been exposed to an antigen, like if you come into contact with a virus or with a vaccine, results in the production of antibodies. Now this provides long-term protection because your body has produced the memory cells against this antigen. So unlike passive immunity, which is where you just get your antibodies from an external source, for example from the placenta or injected into the body, your body has formed the plasma cells and the memory cells, so there's long-term protection as a result. Artificial immunity is acquired following deliberate exposure to an antigen or pathogen. So this is not like you're just walking about outside and somebody sneeze on you. This is due to deliberate man-made exposure, right? For example, a vaccine. That's artificial immunity. Some examples explain the role of vaccination in providing immunity to pathogens. Well, vaccination involves the introduction of a small amount of a weakened version of a pathogen into the body. It could also involve just a fragment of the pathogen or even just toxins produced by the pathogen. And typically this would happen via one or more doses. Typically, a lot of vaccines are 2 doses and you might end up having booster shots to kind of renew the memory cells. So when the antigens from the vaccine enter the body, the antigens, remember, the substances from the pathogen that your body is able to detect. Plasma cells are stimulated to produce antibodies against the pathogen, or just white blood cells or lymphocytes, even that would work in this case. Memory cells are then formed for long-term protection. So as a result, what you have is a significant reduction in the lag time between the detection and response of the pathogen in future exposure. So what this means is that because your body already knows the virus or the antigen, or it knows the pathogen already, your body will be able to detect it a lot faster and respond to it a lot faster. So it doesn't have to take the time to learn it because it already has been introduced to it via the vaccine. And the symptoms of the disease will be greatly reduced. That makes sense. There are several types of vaccines. And I'll list 3 here. You have live attenuated pathogens, which are just weakened versions. Example, this would be measles, and the Sinopharm COVID vaccine was that too. You have inactivated pathogens, which is the pathogens but they're basically dead, and that would be, example would be polio. You have toxins from the pathogens, example would be tetanus, and well, we know now a 4th one here would be like mRNA vaccines, which basically uses the messenger RNA from the virus to build a fragment of the virus or a fragment of the antigen. And an example of that would be the Pfizer vaccine for COVID. And here we have our last question. It's not a tough one at all. They say despite a decline in new HIV infections in the Caribbean, the region still has a higher HIV prevalence than any other region in the world, except for sub-Saharan Africa. What are 3 factors that may account for the continued spread in the Caribbean? I'll list 4 here. There's a lot that you could say. Wherever you say, I just want to add a little detail to it. So it's 6 marks, so you state your thing and then you explain it. Common examples of this would be the window, the time between infection and detection is quite large. It could be several months before you actually know that you have HIV. So if you get tested in January and you have sex throughout February and March, you might know you have HIV until April, and by then you don't infect a host of other people. So the long period of time between infection and detection contribute to large numbers of infected individuals transmitting the disease, even if they have been tested. The second one is increasing sexual promiscuity and having multiple partners also contribute to higher rates of transmission. So, I mean, people could do what they want to do. If you want to have 3-4 people and everybody okay with that and everybody, nobody getting vexed, you could do that. But if you need to have proper protection barrier methods and things. So when this is coupled with limited prevention programs, so like condoms are not available in the area and a general lack of sex education, well, you're running into trouble there if you're having multiple partners. As I say, you do what you want to do, but make sure and use protection. 3. The stigma of HIV infection is still high in the Caribbean, which may prevent at-risk individuals from accessing a clinic to get tested. Basically, you feel you have HIV, but you're too ashamed to go in the clinic. Nobody wants to actually say they have HIV because they're afraid that they don't get a job. They feel people will stay away from them. There's a stigma. And lastly, in some areas, there may be inadequate facilities for diagnosis and treatment. And of course, this would result in less infected individuals being tested and would actually increase the spread of the disease. Other things I could put here is that there's ease of travel. There's a lot of migration between areas. So if you have a country with a lot of HIV infected individuals and they come to our next country, they could easily spread it, especially through sex tourism and things like that. drug use involving hypodermic needles if they're sharing needles, and poverty, which basically, if you don't have the means to access condoms and things like that. So any three of these, once you properly explained, you should be good. And that there's the end of this paper. See you all, yeah?